Sarah Miller here. So this week we'll be looking at an enzyme called catalase, what it does, how it does it, and where we can find more of it. The experiment we'll be carrying out will be using catalase from yeast to produce lots of oxygen to fuel our noodle rocket, which we'll see near the end of our video. First, let's look at the reaction. Oxygen is created as a product of hydrogen peroxide decomposition along with water. The yeast acts as a catalyst for this reaction. This reaction produces heat, which makes it exothermic. Enzymes are common in biological reactions and act as catalysts for the reactions they control. Catalysts are not charged or changed by the reactions they participate in and are not used up by the reactions either. Each enzyme has a specific reaction that it's specialized to carry out. This is because the three-dimensional structure of the enzyme is suited to attract and hold only specific molecules in its starting position, called the active site. The molecule it holds in its active site is called, the molecules it holds in its active sites are called substrates. The initial energy that must be available in order to break the bonds of the reactant molecules is called the energy of activation. Enzymes are beneficial because they lower the energy of activation. While the conversion of hydrogen peroxide into water is catabolic, the rate that it occurs is very slow. Because hydrogen peroxide is toxic to living cells, the catalase enzyme is made by almost all living cells that are exposed to oxygen. So when you fell down and scraped your knee when you were a kid and then treated the wound with hydrogen peroxide, the bubbling was the oxygen produced by the catalase in your cell. Hydrogen peroxide is actually toxic to living cells, so it kills your cells and bacteria alike. Non-enzymatic things that can affect the progression of the breakdown of hydrogen peroxide are light and heat. This is why the bottle is dark brown and recommends that you store it in a cool place. And it's also why there's an expiration date. A couple of things that can affect how fast an enzyme reaction occurs. A couple things can affect how fast an enzymatic reaction occurs. This includes increasing the amount of enzyme, which will increase the reaction rate. So adding more yeast to the hydrogen peroxide would speed up the decomposition of hydrogen peroxide. Another factor that affects how quickly an enzyme works is temperature. Increasing the temperature to a certain point will make the molecules move faster, which means they'll be more likely to engage in a chemical reaction. But once a certain temperature is reached, however, the hydrogen bonds in the enzyme's protein structure begin to fall apart in a process called denaturation. Catalase begins to denature at temperatures over 37 degrees Celsius, and the enzymatic capacity begins to decrease also at this temperature. At approximately 50 degrees Celsius, the enzyme becomes completely denatured and there is no reactions occurring at all. The type of catalase present in yeast is the heme-containing enzyme, which is ubiquitous to most aerobic organisms. And aerobic means they use oxygen for their metabolism. We will be focusing on this group of catalases for our discussion, although the structures of 16 variants of the catalase enzyme are known in nature. The overall heme-containing enzymatic reaction occurs in two distinct stages. The first stage involves using the hydrogen peroxide molecule to oxidize the heme and porphyrin ring and creates an iron 4 species and a porphyrin cation radical intermediate via single electron transfer and one water molecule. In the second stage, the radical intermediate is reduced by a second hydrogen peroxide to regenerate the enzyme and its resting state. and also produces one water and one oxygen molecule. So that's the catalase enzyme and the reaction that occurs during the degradation of hydrogen peroxide. Now let's see this reaction in action. Okay, so here we have all of our ingredients. We have hydrogen peroxide, a teaspoon, platinum, activated yeast, and ZT pasta. I also got some rigatoni pasta, which is a little bit fatter, just for fun. And then we have our mason jar with a hole in it with a ZT pasta sticking out of it. Then we have our lighter, and yeah, we're ready to go. Okay, oxygen. Okay, so we have our hydrogen peroxide. This is take 3. And we're gonna add a teaspoon of the instant yeast we have here. And we're gonna put the lid on, give it a little swirl, and hold the top this time, like we're supposed to. And then start this thing on fire. There we go. All right. Got ourselves a little rocket. Pretty cool. All right, that works.
We are doing 30% hydrogen peroxide. So we've got that in here with our dish soap and our color. All right, now I'm not crazy, so I'm going to step back from here while you do this.
While I do it.
All right?
Thanks.
So we're going to count to 3, and you're going to pour that in there, and then you're going to step back.
Okay.
All right, you ready?
I'm ready.
All right, keep your eye on the gigantic flask.
Okay.
All right, here we go. Ready? 1, 2, 3. Pour. Step back. Oh, look at that foam. Now that is science. Wow, look at that. And you see it's steamy?
Yeah.
That's it's an exothermic reaction, so it creates heat too. This one you can't do at home. These two you can, and we'll have the instructions on the website for that.